We've had a very eventful year. Not all of it good, but a lot of it that I hope to tell you about today has been good. And so I want to thank you all for joining, and I'll try to leave some room at the end so we can have a little conversation about, maybe the up and coming annual meeting or other things that might be front of mind for any of you. And with that, I will go ahead and jump in. So we had many site visits this year, but the one that was the official site visit was just recently, and the outcome was to continue support. This is what they call a high bar review, and it doesn't get any better than that. You can see there are 3 levels. There's a mid-level and a low level. So we are at a high bar for the second time in a row, and this is a very good thing. So thank you all for your hard work making that a reality. Some of the things that they noted on their review, which I just want to share with you, is a couple of significant achievements up front, that we've created a convergent cryobiology scientific community. I believe that as well, and I'm very proud of all of us for doing that, along with our collaborators in the Society for Cryobiology and far, far beyond that. We've shown the ability to do human organ scale vitrification and rewarming. We've scaled up coral sperm cryopreservation in the semi-automated workflow to support biobanking and aquaculture. Automated multithermic perfusion platforms with diagnostics. A long-term repository on the moon is our bold plan, thanks to Mary Hagedorn. And we have a cryo foundry, which I'll tell you a bit more about as a vehicle for commercialization. So these were called out by our reviewers, not by us. And so I think that's nice to see that they are aware of what I too find to be significant achievements. And so I just wanted to share that with all of you. We have had a great couple of years in terms of recognition of our leaders. And I'm sure there are more here that I haven't been able to get onto one slide, but I wanted to make sure that we do recognize that we are blessed with a lot of wonderful people working in our community. We also got, as a team award, the Edward Grug Interdisciplinary Team Science Medal in Bioengineering this last year. Lots of promotions and appointments, 40 plus medals and prizes. And when we get into workforce development later, I'll mention, I think it's 14 of our trainees now in academic positions, which is amazing, and even more in industry. So our numbers and metrics are amazing. We are doing very well by any estimate of an average ERC. I think all of the feedback we're getting has been positive. There are some frustrations sometimes in the reviews because you hear them saying, well, can you focus, but can you do more? And I think it's sort of what the NSF does, what the site visit committee does, is really try to see how much further they can push us. But there's no doubt in my mind that we are an extremely high functioning ERC, and we are doing a tremendous job. And I hope we all can feel that and appreciate that with that, especially that last site visit review. So our vision is unchanged. To stop biological time, to advance healthcare bioconservation and food supply and sustainability. You can see many of the applications called out there in cell therapies, translatable tissues and organs and organoids, which is in the healthcare space. We've recruited new talent and leadership. You'll see also new folks talking in this area in the annual meeting. Coming from Mayo Clinic, coming from other ERCs. So our healthcare area is doing well. And in fact, we probably get most of our funding from the healthcare space. But that then allows us to move some of those great breakthroughs into bioconservation and food supply and sustainability, where we're also making some great strides in biobanking of species, coral conservation, vertebrate and invertebrate model systems, space, as I mentioned, agriculture and aquaculture, in food supply and sustainability, food conservation, and invasive species management. So our strategy in the first 5 years was really, we weren't sure that we were going to be able to hit all of our milestones, but in fact, we have largely managed to achieve the milestones that we set for ourselves, that many of them were even into the next, the 5 years, the second 5 years that we're in now. And so those fundamentals were really achieved. Those technologies now in a large extent kind of exist. And so our proposed 6 to 10 strategy is to really begin the process of translation. And I think you'll see, as the site visit team did as well, that we are on our way to translating these technologies. And of course, ultimately, we want to sunset, but sustain ATP-Bio, and that is what we would have in year 11 and beyond. And we'll talk a little bit more about that and a new committee and new effort that we have to try to structure ourselves for that time that will be coming faster than we all think. So imagine that we could have organs waiting for patients instead of patients waiting for organs, and turn, transplant surgery into a day job. And imagine if we could bank cellular therapies and organoids and bring them to bear for patients when and where they were needed, such as pancreatic islets to cure diabetes. And imagine if we could preserve food longer and feed the hungry world. And imagine if we could create a lunar biorepository to actually preserve life itself, keystone and precious species, where we don't require liquid nitrogen or an electrical outlet on the far side of the moon. That's what we are working towards. And these key achievements that we've already brought to bear in the first 5 years are going to make those possible, especially as we translate them. And you can see here, we're doing that in all of our test beds. So in organs, we've taken multiple high-level journal covers with our work. We're being featured in the lay press, the New York Times, Forbes, etc. In cells and organoids, publishing in some of the best journals out there on multiple different systems, from red blood cells through islets, microphysiological systems, and more. And in organisms and food, also many high-level publications in Nature Com, Advanced Science, and Genetics, talking about all the different test beds that we've been able to cryopreserve that we weren't able to cryopreserve before, and then also in the far right, the moonshot to preserve endangered species. And that's actually an interview with Mary that just showed up in Nature, which was really fun to read about. So in our early years, we were 5, and then we became 6 institutions, and we have had activity at all these institutions. And we had thrust areas instead of what we have now, which are integrated thrust areas. But now we've significantly grown, and we've added quite a bit of new investigators, associated projects, people that are working in AI, ML, and things of this nature, which I'll say more about. And we are really building out this convergent cryobiology network and team that the site visit team also noted in their review. And we're also adding new efforts in ethics and public policy and workforce development, as well as innovation, that you'll be hearing about in addition to our convergent research. So one thing that's important when we talk about building out this ERC is making sure that we're not just working at any one institution, but that we're actually working between institutions. And so this is really tracking that and showing that indeed we do have multiple projects that are working across our institutions. And this was both in years 1 through 5, but also now projected into our renewal years 6 through 10. So now going through some of the pillars, I'm going to just walk through some of the highlights with you. And what you'll see here is that we've made significant progress across all of our test beds. And this is, in the organ space, 10 days of high sub-zero pig kidney preservation from the MGH group, followed by a transplant that was successful, and an acute transplant where the kidney was able to make urine. We've also had a 100-day vitrified rat kidney transplant, and we're now vitrifying pig kidneys successfully. And then clinical studies that are ongoing in the high sub-zero organ space, isochoric approaches in particular, which look like they may in fact be translating faster than any of the other technologies, which is extremely exciting. And they may also help us move through the FDA in the best possible way, where they could blaze a trail and help the rest of the technologies move. Lots of startup and licensing and IP going on in the organ space. In the cell therapies and organoid space, we have our result that we published on islets that we've now been able to scale to clinically relevant IEQs, so 700,000 islet equivalents that we can put on a single mesh, which is amazing. And obviously with the ability to cryopreserve with high viability and function, that means we can also do pooling of islets if we're talking about isolated islets, or we can take stem cell derived islets and put them all on a single mesh. Lots of other activity going on, including non-human primate, both islet preservation and transplant, with some very exciting data going on there. Some new funding in the area, new startups and licensing in the space, and Army and ATP-Bio are on each other's membership now. In the organism space, we have a number of grants in model systems such as Drosophila, zebrafish, but we're also working on mosquitoes and coral with a variety of our ATP-Bio partners. And lots of workforce development now going around approaches to train in these protocols. In particular, Drosophila, we have a Canvas website. We have a workshop that we'll be offering this spring. Our group with Tom Hayes in the lead is going down to the Drosophila meeting this year, and we'll be recruiting people that want to actually do this for the first time. And because this is a little bit ahead of what we have with some of the other protocols, we're hoping to use this as a model to build out other protocols around zebrafish and some of the other important organisms and other test beds that we're working with. With biorepositories, we are collaborating with a number of different groups, including the Smithsonian, and thinking about this lunar biorepository, but we're also engaging with TAMU around space and food. And in total, we're making tremendous progress across all of these test beds. So one of the exciting things, and this is really, we were getting a lot of feedback from the site visit team, so I think we do need to give some credit to the review process itself. They've been noting, which I'm sure we all were noting, that AI and ML is here, and it's really playing a role in pretty much every research space now. It's in all of our lives, whether we like it or not. And we now have consciously and explicitly written this into our integrated thrust areas with a variety of new AI ML experts that you see on the top there, which is in each one of the different integrated thrust areas. but also we have a number of our junior faculty using AI/ML in their grants, such as the NSF Career Award that Becca got just recently, but also in their projects and their efforts across multiple institutions. And then also we have a number of associated faculty that are working in the AI/ML area. And just this morning, I was on a call with Alan Roman and Matt Palpam, who had not met each other before. And we're thinking of putting together a physical characterization study. And one of the reasons for that is because the uses of AI/ML are dramatic. So we have, with automation and the ability to create, let's call it automated labs, we now have the ability to get tremendous amounts of data, and we can use AI/ML to guide us to build better high throughput screening, CPA cocktail optimization approaches. And this may be more on the toxicity side, which is the interest of some, or it could be more on the physical side, which might be more on the glass and viscosity side. Ultimately, they'll need to come together, but the ability to use AI/ML for that, I think, is compelling and of great interest to the community. Also, multi-omics-based optimization of storage conditions, another opportunity for AI/ML. We're using machine vision, and with Swati Gupta joining us in ITA3 from MIT, she is focused on fair allocation of rare and precious biological systems as an example. And so really opportunities across the ITAs and across our projects to integrate AI/ML to do more. So this is sort of our classic technology slide. Organs are an example, but the concept that you can store at high sub-zero or go to low sub-zero temperatures, such as vitrified, this is really what has been driving our breakthrough technologies. And we literally have breakthrough technologies for each of these: supercooling, partial freezing, isochoric, and vitrification. And around the organ space alone, there's a lot of IP generation, licensing, and startups. And as engineers, you know, the ability to manipulate the temperature, pressure, and concentration to achieve this lowered temperature preservation, lower metabolism, and therefore longer storage is really what drives our ERC. And so in organ cryopreservation, since our inception, we have had multiple breakthroughs and multiple scaling up, if you will, from rodent systems into pig into human. And this is extremely exciting because every one of these technologies, as I mentioned, is moving towards a translational opportunity or a commercial opportunity with some of our partners on the right. So vitrification, partial freezing, isochoric, all of them are showing, I mean, there's tremendous excitement out there about what is happening in the organ space. And so this is going on also across our test beds. We're deploying these different breakthrough technologies, and they act as model systems for research and development, but they also themselves each are products. And of course, this ends up being, as I mentioned before, probably driven first by our ability to get the most funding so far in the healthcare space, but then it's transitioning into both biodiversity and food and sustainability applications as well. And you can see our new integrated thrust areas there: biostabilization engineering and ITA2 cryothermal processing, which would be both cooling and rewarming, and then the cryo supply chain management, which is a completely new ITA, which is in response to this need to translate and to get our breakthroughs out into the world. ITA1 biostabilization engineering is really how do you prepare your system to go into a state of biostasis. So CPA discovery would be one example, but gene therapy and other approaches to build in the ability of the system to survive biostasis would be others. And so we're using, this is just a showing that we are not just moving forward with organs, but also in organismal. You can see both technologies on the bottom there. that are many of them published and have use, as I said, in healthcare as well as in bioconservation, and then all the different species that we've worked on above the timeline, and then working towards translation and use with vertebrate or invertebrate model system centers like Zebrafish International Research Center or Bloomington Stock Center for Drosophila, but then also smaller companies that are working on other approaches where bioconservation or food is important. And you can see directly in food that we're also making dramatic headway. A lot of this is coming out of UC Berkeley and TAMU, the work of Matt and Boris and others in terms of reduction of microorganisms and different ways to impregnate or bring nutritional fortification into plant materials, whole milk. There's a startup, NutriMilk, that actually went through our cryo foundry, and then protein sources and feed, et cetera, on the bottom there, having to do with 3D printing and other approaches that are important in food, and even freshwater and saltwater rotifers, which are like the first thing that an aquaculture species might eat once they started to grow. And again, moving to the right towards translation with multiple different partners. So we have a revised three-plane diagram, again, being in this moment where we're in our renewal now officially in year 6. And we have those integrated thrust areas that I just introduced. We've consolidated our test beds from 4 to 3. So now on the top in blue, cells and organoids together. And that kind of comprises in the healthcare space, that really is cell therapies in some sense, are both cells and organoids, organs in the middle, and then whole organisms, all three there. And then our translation for impact on the right, our pillars, as you see them there, and then working towards financial sustainability, which we'll talk about a bit more. And then also stakeholder engagement to make sure that our translation will be successful. And there's a fair bit more to say about this, and a lot more engagement needed around that. So we have new milestones. As I said, we were able to achieve many of almost all of the milestones that we thought were going to be the milestones going through the whole grant, the 10-year grant. And so now we've come up with new milestones, having achieved basically all of the milestones that we set for those breakthrough technologies that I already mentioned to you in terms of scaling and ability to work on different test beds. And so here, now we've integrated in artificial intelligence, for instance. So we are actually optimizing CPAs, alleviating injuries that we know occur around endothelial and mitochondrial injuries, and scaling up and using advanced machine vision and other quality control approaches, bringing in automation, integrating storage systems, and then ultimately getting out to the cryo supply chain and managing that. And that is with and needs to be with our stakeholders. And so figuring out how to work with end users, patients, broad public, surgeons, et cetera, and engaging regulators to ease this translation is becoming really important. And then figuring out which products are going to go first. As I mentioned, I think isocoric may be leading the charge. They seem to be getting into the clinic and working well, and they do not have some of the barriers of some of the other technologies. So maybe they will lead, perhaps, and they will help blaze a trail, we hope, for some of the other technologies. So we have all of these new projects. This is really just saying or showing the methodology that we have to populate our different integrated thrust areas with our test beds and projects. And rather than walk through all of the different projects, I'll just point out that in the bottom right, intentionally under ITA 3, And not just in the bottom right, but a lot of this is open and evolving, especially around ITA3. And as an example, we're trying to figure out how do we do shipment. And so, for instance, we just shipped Drosophila larva from Minnesota to Purdue, where Lizanne now is a junior faculty and one of the trainees of ATP-Bio. And he was able to successfully grow up some of those larvae and also send back this dry shipper that had larvae in it. And we are now growing out larvae as well. So these kinds of examples of what is possible in terms of shipping our preserved test beds will be needing more and more of that. And that really does fit well into ITA3. So in ethics and public policy, continuing work there around the Net Ethics Project, which is really studying ATP-Bio as a laboratory of these network level research ethics teams. There was a whole Net Ethics Day. It was in November. Susan, Tim, and others really hosted. It was a nationwide Zoom meeting that looked across not just ATP-Bio, but even other ERCs, and began to involve them in this really important conversation about how do we best do team science. And then continuing collaborative research and publications. I'll be highlighting a few of those going forward here. And then anticipatory governance. We just had a webinar where we began that process actually with Susan Alpert. We had already had some conversation with her, as well as I and Mehmet have been talking with Dev Srivastava, who's an FDA expert in thermal performance. And Michelle Tarver, who is the head of CDRH, is actually coming to the annual meeting. She'll be giving the lunch talk from the FDA, and that'll be in the April 22nd. So the proposed work in ethics are surveying and analyzing the views of stakeholders. So as I said, this is going to be a huge and important part of what we need to do going forward. Engaging external stakeholders and talking about bidirectional exchange and learning from and talking to both patients and surgeons, which has already begun, thanks to Susan and Tim. Advancing scientific field of biopreservation, and this is through collaborations such as this harmonized terminology paper, which just came out, and a responsible translation of our technologies, regulatory governance issues, and of course this consulting with other ERCs like Susan and Tim did for that Net Ethics Day. That seems to be continuing through SEEK, which is a new stakeholder engagement initiative at the NSF, which Susan is actually chairing. So this is just hot off the press, so I'd let everybody know. Laksha was leading this, and Susan anchoring it as the senior author, and many of us participating in the need for harmonized terminology and cryopreservation. And this is a great example of how we can do more together to actually make people aware of what we're doing and not get confused with the terminologies that we use. Lots of other great cross-disciplinary dissemination already underway with Susan, but also with others. And we have, I won't go through the whole thing here, but I think we're going to be, I was invited just recently, well, perhaps to be giving a TED Talk in Minneapolis around what ATP-Bio. I know that there's also an invertebrate cryobiology manuscript for one of these NIH workshops. There were 6 of them last year around cryopreservation. Lots of different webinars, both ours as well as the Society for Cryobiology. We're doing them jointly. A lot of really great opportunities for us to get the word out, and also to make sure that it's being done in the right way to engage the stakeholders as well as our community. And I'll highlight there over 1 million plays from Shannon's "Can You Freeze Your Body and Come Back to Life?" That is the way you get impact, is making sure that people can see what you're doing. So by level engagement strategy around organs, there's level 1 and level 2 on the left there. And There's upstream engagement, which we have been encouraged to do, and I think we are doing. But there's also the opportunity, and we've heard this now from some of our industry colleagues, that we need to do that very carefully because we don't want to actually get in the way of what they want to do, which is a product-specific engagement around something very specific, like cryopreserved organs or cryo devices or modified organs. So this is a very important ongoing conversation. This was part of the January 2026 EPP-IE meeting that Susan and Tim put on, and we had 3 great panels with industry stakeholders, regulatory stakeholders, and clinical and patient stakeholders. It was a really informative meeting. And Tim's paper definitely came up quite a bit, and this is a great way to show how we can begin to start these conversations that need to be had around how we're going to do this. How do you govern these new technologies around ATP-Bio? So in the innovation ecosystem, which collaborates strongly with EP3, especially around stakeholder engagement, we've had a lot of activity. This is Dan Chen's work, along with Ken Rosen and Ikram and Carla. So we have 44 members, many from industry, but some from government and as well as innovation sectors. 9 patent applications, 1 licensing agreement, and several are underway. Even today, I was having some email around one of those that is looking very promising. And our TRL levels are increasing in all of our projects. And we have really invested in and grown startups. So we have had 8, and we're expecting more. And then this, as I mentioned, it's accelerating cryotechnology through stakeholder engagement is something that IE is an innovation ecosystem is doing together with EP3. And this is, going to be a broad effort across industry policymakers, healthcare providers, and conservation leaders. We've got, as I mentioned, 44 members, as you can see, spread around. And we have this robust startup ecosystem, and you can see those on the right, but also lots of other groups that we're working with that help. And we've got many of our IAB members are generously spending their time actually working with us on a new effort called Cryo Foundry. I'll tell you a little more about that actually helps mentor some of the younger folks, many times our trainees, that are thinking about actually doing these spinoffs. And so this actually leads to this really amazing, I'm amazed, commercialization pipeline approach, which Dan has put together with help from his IE group. And you really start with commercialization assessment, and then you go into this Cryo Foundry, which is really kind of like a mentorship group, where you're getting a lot of feedback from some of our IAB members. They help you with business plan development and either formation or licensing. And so there's, I wish I had seen something like this when I was younger, because there really is no mentorship, or I have not experienced the mentorship in my career as an academic, but we are really putting it together here in a really positive way. And the discussions and the accolades after we have a cryofoundry meeting from everybody is just so gratifying to hear, because you're hearing it from everybody. You're not just hearing it from, the people that are starting the companies and saying that the trainees, that was really good, that was really helpful to me. But you also hear the IAB members saying how much they're enjoying it, how much they enjoy giving back and working with the trainees. So it's just a really wonderful thing that we brought into existence here that Dan and the IE is doing. In workforce development, we have been doing a fantastic job. Jennifer McCambridge is our new program manager. And Rhonda Franklin, of course, is leading as a faculty lead. We have continued our exchange fellowships. These have been really impactful, and we continue to make them available so that people can move between sites and create some of these projects you saw on the national map so that we have collaborative efforts. We've had over 145 undergrads that have worked through our research programs and 50+ grad students and postdocs and faculty members that have mentored some of these young researchers. We are building out courses. There's one that's taught here at Minnesota that Joe Congas and I were teaching, but there are others being developed. Matt's developing one or more down at TAMU. And we also have new, these actually they're not new. The older lab skill videos have just been loaded. Ellie was telling me those just went up online on our website and I think on YouTube. And lots of opportunities now for curation and to use these different videos to achieve different things. And just as one idea that was kicked around, imagine a sophomore or freshman freshman seminar where we could use something like Paul Izio's really wonderful talk that he gave at the Texas A&M annual meeting. Maybe it's a 15-20-minute talk about hibernation, bear hibernation. And you get, so you have a world expert on hibernation like Paul giving a talk, but then you allow that to be just a part of a course where I or somebody else who's not an expert in hibernation could just have a discussion with undergrads about, what is hibernation, maybe use a film clip to remind them of something they saw in a Marvel, one of these Marvel films about what, Captain America going into the ice or something like that. You stimulate that, then you watch that real deal with Paul Izio talking about the bears, and then you have a follow-on conversation. And then you do that again and again over 10 or 15 topics. All of a sudden, you've got a course that could be disseminated across any of our institutions or even across the world. STEM outreach has been a big effort, and Catherine Harrimans has been doing a fantastic job. Just did it last week, I believe, again at the Learning Center for the Deaf. And we have other efforts at many of our other institutions. So we've got a lot going on. Postdocs to academia, it was 14 plus 3. Postdocs to industry, 14. PhDs to STEM industry, 22, and law trainees as well. So, just an amazing amount of workforce development, amazing amount of trainees that have gone out and are doing great in the world. Total number of trainees is 337. Strong transition of the postdocs to final career goals, as I mentioned. The STEM industry is a critical destination, and we're tracking all of these folks, which is great. And this is really, one of the biggest things that we should be proud of, I think, is that we have trained successfully so many wonderful young people. Our strategy is really, inspiring and sparking interest at a young age. We're collaborating now with MESA, Opportunities Industrial Centers of America, OIC, as well as the Youth Scholars Program and some of the work that Catherine's been doing. We're preparing and building knowledge and career pathways that's more in the undergraduate and graduate area. And then advancing this and developing leaders. And this would be particularly through our Scholars Leadership Council, which is led by Alexandru Ulate this year. And Russia is also there. This is something that we just signed on to, this OIC of America partnership. Amy Klobuchar actually knows Louis King. So Louis King is the president of OIC of America. And we were here in Minnesota celebrating our partnership just not a few months ago, and Amy actually didn't come directly, but she actually, it was a very nice video that she made. She obviously knows Louis well, and we're hoping to build this out. Jennifer's in talks with Louis's team about giving an ATP-Bio STEM Saturday on October, And we're going to have a STEM Fest event in June, which will be featuring some ATP-Bio relevant curriculum as well. Short courses. This is something that actually will be important as we start thinking about sustainability, because these short courses, if done well, can actually be a great source of funding, and particularly if you have practitioners that are taking them around, let's say, organ transplant, like a perfusion short course. But the Drosophila course that we've developed here at Minnesota is probably the first one that's ready. And as I mentioned, we're planning to do a workshop, multiple workshops actually, this spring and summer. So if you know anybody who is interested in actually transitioning some of their fly stock, instead of keeping them alive, flipping them, or trying to send them to the Bloomington Stock Center, we can help them actually freeze them down and put them away in a tank. And I've heard stories recently, Tom Hayes and I were talking about this, that there are actually groups now that Bloomington is turning their lines away. They don't want to take any more lines. They're full up. And so I think cryo is going to become increasingly important to groups like that, especially, but even also to Bloomington that doesn't want to keep, the 75,000 or however many lines that they have, half of more of the lines in the world going. Organoid training and zebrafish, these are all different ideas for short courses in the future, and I'm sure there are more. And this is an important future goal for us. So that brings us then to our sustainability plan. And this is a slide that Mehmet put together, obviously with a lot of input. We want to be thoughtful about, not just relying on one thing. Membership dues are great, and we appreciate our IAB members, and this has been growing. But we're going to need more than that to keep us going. We're going to need target annual amounts of 650K or above to sustain our core operations. And that means essentially 4 to 5 full-time positions, especially staff, is what we're talking about here, to maintain us as a group. And then hopefully we can go above that 650, and we can use that for seed grants and junior faculty support, continue some of our exchange programs and things of that nature. What you can see there is that we've actually achieved, many of these things there. You might say, well, that's not maybe realistic, but we've already pulled in $1.4 million of philanthropy, of institutional support, and we have a 20% cost share. There's been 400K that came into the MGB MIT fund, and we've got 1.2 million of philanthropy already. And our member fees are certainly a wonderful source. And you can see our strategy there on the left is like, we need to keep all of these things, the network and governance. This is what the staff would be doing, the workforce development, and then the research, innovation, and broader societal impacts. Keeping all of that going, we need to retain our administrative core. And I will then reflect on our mock site visit, which actually set us up really nicely for the actual site visit that we just had that went so well. By the way, there was no, overnight questions or anything like that. We got it done very in a streamlined way. And one of the things that came out of the mock site visit was that we all sat down towards the end when Mehmet had actually, I think Mehmet presented this slide. And I'm not sure if he actually talked to Mesh first or not, but he volunteered Mesh, and Mesh agreed to be the committee chair for a new sustainability committee. And we are going to start putting some flesh on the bones of these ideas here to really make this sustainability a reality. So I thank you to Mesh for doing that, and I'm sure he'll be calling on many of us to participate. I wanted to make everybody aware of our new website. and our annual reports. These are thanks to Ellie. They're up online, and you can pull them down. These QR codes should get you there quickly if you're looking at the screen now. And I want to invite you, as always, to engage with us. If you're here, you're already engaged, but there are many other ways. We have a public webinar series, and we have on March 3rd, the State of Research. This will actually be ITA trainee co-leads, I understand, will be presenting that. And I will mention that we just had a webinar that was entirely trainees, 3 different trainees, and they did a wonderful job. Very, very interesting and engaging work, state-of-the-art work coming out of our labs in ATP-Bio. And also on the 17th, March 17th, the Lunar Biorepository will be presented by Mary Hagedorn. We have an ongoing ATP-Bio Trainee Tuesday, which Alexandra Ulate can tell you more about, or Jennifer McCambridge. And then I wanted to make sure to invite everybody to come to our ATP-Bio annual meeting. And this will be April 20th to 22nd here in Minneapolis. And just to give you a quick preview of that, we're going to have on Tuesday, April 21st, from 10 to 11:30, Eric and Seb, one of our ATP-Bio partners, will be talking about advancements in organ transplantation. My understanding is that's going to be many of our translational partners are going to be presenting from companies. And then advancements in preservation for cell therapies will be coming after that, 1:30 to 3. Mehmet and Uzair Rajput, one of our IAB chairs, our IAB chair now, actually. And that'll be very exciting. I think we have Julie Alexson from Mayo will be there, as well as Jonah Temeroff, who's the director of CMAT, which is another NSF ERC in the cell therapy space. And then we'll have a biopreservation technologies panel, which is coming together. And then finally, a DMD reception and student design showcase with our poster session. That'll be from 5:30 to 8 on the first day. The second day, we're going to have technology advancements in cryopreservation. Matt is going to be leading that with Rob Goldstein, also an IAB chair. And that will be featuring many of our trainees and some of the wonderful work that they're doing, and somewhat shorter talks so we can hear about more things. But I'm really excited for that session. As I mentioned, it's not shown here, but the lunch talk in between will be from Michelle Tarver, who is the director of CDRH at the FDA. Super excited to have her come and talk to us. And then 1:30, we're going to have a biopreservation technologies fireside chat. Gloria Elliott, who is a program manager at ARPA-H, and she's leading this new BOSS initiative, which is on desiccation preservation of cells, will be coming. She also oversees other programs that are in the more complex tissue space and organ space, like 34 Lives. So I'm really looking forward to that fireside chat with her, and then we'll be wrapping up the annual meeting after that. So with that, I want to thank you all for coming to this State of the Center. I'm going to stop sharing and look forward to any questions or discussion that you all would like to have. You didn't expect me to stop that, huh?
We did not, John. Thank you.
Certainly.
And I did ask Mesh before I announced his willingness to help.
Oh, that's good. Yeah, well, I'm not sure if he's on right now, but I thought
he was on. I saw him. I don't know
Okay.
if he's on now. Yep, he's here.
I see. Yeah, I don't know if Mesh wants to say anything about sustainability.
What's our biggest hurdle going forward?
Our biggest hurdle, well, will we be funded in the fall? Who knows?
Yeah, I mean that goes without saying, but I suppose the technical scientific goal.
Yes, that's a good
I
question.
mean, Alyssa and I have been meeting offline. She's my co-chair, or I'm her co-chair, depending
All
on how
right,
you view it. And so we're cooking up a plan that we hope to present in the annual meeting.
All
So
right.
that's what we're doing behind the scenes.
that's
And,
awesome.
you know,
Thank you.
I would add a box to Mehmet's slide in terms of education, because I think,
Education. Okay.
yeah,
And that you mean that in the sense of short courses or beyond that too?
beyond that, I mean, short courses are one thing. We discussed an idea about engaging actually high school students in a camp-like structure where I can guarantee you that parents who are interested in students going to elite colleges would be willing to pay for this kind of thing.
Yeah,
Okay.
that's a
So,
good idea. Yeah, you've done those camps quite a few times in the past, Mesh. So
yeah. So I think that would also be it'd be a good thing, and it could engage everybody from, fellows to, undergraduate students, graduate students, postdocs, as well as faculty.
That's a great idea. Thanks for that. Yeah, so I mean, I'm sure we're all looking forward to hearing more at the annual meeting. And thank you and Alyssa for your work on that.
Sure.
So,
We have
Emmett,
a couple of questions in the chat here.
sure, you want to go ahead and hit them, Ellie? I can't see them.
Yeah, sure. Raphael was asking, can you speak more about how isochoric vitrification has advanced in the last 5 years, and what do you expect to see in the next 5?
Yeah, I am not somebody that does isochoric vitrification myself. I think it is, I'm not sure Matt is on or Yoed, but they have both tried that at different times, so I can give you my very high level. If you try to vitrify an so you have a liquid system and you fill it to the brim and get rid of all the air pockets, and then you try to reduce the temperature and any ice forms in it at all, you'll spike the pressure in the system. And so that's been a problem with isochoric vitrification, because basically you will end up quote unquote vitrifying and getting to very high pressures in whatever the remaining liquid is, which ends up being amorphous if you go deep enough in temperature. But you will also have, spiked the pressure so much that it's actually bad for the biological system. So that's my high level answer to that. I'm sure Matt would push back or Boris would push back on me. I think that's what happens.
Yeah, assuming the question was very specific to vitrification, but isochoric is now being used for other biopreservation approaches,
Correct. Yes.
not vitrification, but supercooling and cooling, so on and so forth. And it's showing significant effectiveness in those areas.
Yes. And so that's where, Raphael, we talk about high sub-zero and low sub-zero. And so vitrification tends to be below the glass transition temperature, kind of like by definition, which, tends to be lower, minus 100 C or even lower than that, depending. And, Greg's got an answer too, but Greg, we'll come to you in a sec. But as Mehmet was saying, there's now some companies that are doing isochoric supercooling, so no ice in the system at all, and they're seeing significant increases in the ability to preserve organs up to days in some cases, and that's pretty exciting because that could be something that goes to the FDA.
Seb online, John. No, Seb wants to chime in.
Okay, I think Greg had a quick answer.
Greg first,
if you
yeah.
wanted to answer, and then Seb.
Do you
Greg, are
see?
you able to come off of your mute, or do you want me to?
There we go. Hi, John. Yeah, I've just, as far as the isochoric vitrification thing is concerned, anyone who's watched anything vitrify will recognize that there's a vast reduction in volume caused by thermal contraction as the system gets colder, maybe 20% volume reduction, something like that, maybe more. Maximum ice formation in pure water is like 8% volume increase. So any attempt to vitrify something isochorically is going to have a pressure decrease issue, not a pressure increase issue. So I just have to correct you on that point.
Thank you, Greg. I appreciate that. And I see you had a different question about the pig kidneys.
Yeah, I was very interested in what you said. I gather, so I haven't kept up on this literature, I'm embarrassed to say, but maybe it's partly because there's so much of it. But so I congratulate all of you for that. But
It's a good thing.
you mentioned successful or some success with pig kidney preservation. I thought you were referring to vitrification at the time, but later I saw something about a 30-day preservation success, maybe with hypothermic preservation. Maybe you could just comment on that.
Yeah, this is actually MGH's work. So it's Korkut's work. I don't know that Korkut was able to join us today, but Mehmet, did you want to? I mean, I can try and do it, but you may know more.
Yeah, I mean, that's the partial freezing. I assume that's what you refer to, John, and
Yeah.
where we could reach, partially frozen means, those of you who are not familiar, you're in thermodynamic equilibrium because you have ice, but you try to form the ice in vasculature in places where it won't cause damage to organs. So you could go to a little bit deeper supercooling temperatures and to -10, -15 degrees Celsius, but in thermodynamic, the stable state. So in those cases, we have not done transplantation yet, but based on the typical clinical criteria used clinically to release an organ, it's doing very well.
Okay, thank you.
Great.
Just a
Seth,
quick,
did you want to? Yeah, go ahead.
yeah,
Sorry.
just a quick comment on the isochoric vitrification. Definitely, Matt and Boris should talk to it. But I think, Greg, what you're saying was true, and then they succeeded in demonstrating that you can actually dial in the characteristics of your CPA, so that if you want, either contraction or expansion, you can kind of dial it up and down. And I think that's in that Nature Comms paper I just pasted. But that was in the context of coral, so I don't know the details.
I'll have a look at that, but all aqueous solutions are going to contract thermally. I don't see how you get around it.
Okay, I actually, that peaked. So I think it's whether it contracts more or less than the chamber, right? Because if you design your isochoric chamber so it also contracts slightly with cold,
Oh,
I think
well,
that's
yeah,
what they
that
did.
would-
I think they found a way to balance the
I do-
CPA and the chamber.
yeah. If you inject volume or allow volume to decrease in the system, then it's not really
Constant volume.
isochoric, is it? Yeah, it's
It
not
is
really
not
isochoric.
isochoric.
So, yeah, it's kind of a cheat, but anyway, okay.
Well,
So-
it worked to have surviving vitrified coral. So I think that's
Yeah,
the most
well,
important.
No.
Okay.
I don't
Okay,
argue with
guys.
success. I argue with terminology.
Brian
right.
has a question, I think, related to that.
Yeah, and then I think we might need to wrap it up because we're getting to time. Brian, go ahead.
Supercooled water with no CPA expands with supercooling. There's a threshold concentration of cryoprotectant below which you get pressure increase with supercooling, and it promotes ice avoidance, and above which you get this volume contraction and pressure decrease effect that promotes ice formation. And I think the coral work was near the threshold of that, perhaps. But this is why you're seeing so much more success with isochoric approaches to supercooling than you are to, or that you would see for isochoric vitrification with the high CPA concentration necessary for organs.
This is a great example of why we need Greg and Brian on our ATP-Bio Partners membership group. So Dan Chen will be reaching out again. Gentlemen, we hope you can join us.
We'd love to, John. Thank you.
Okay. Thank you all. Thank you
Invoice
all
will be in
for
the mail.
coming.
Okay.
Yes,
Thanks, everybody.
thank you all for coming. Really appreciate it. Looking forward to seeing many of you, I hope, at the annual meeting. Reach out anytime, everybody. Take care. Thanks so
Thank you,
much.
John.
Thank you, John. Great show.
All right. Thanks, everybody.